Today we will discuss how the destruction of the RBC. So the average age of the RBC, that's the red blood cells, red blood corpuscles. So RBC normally lifespan is about almost 120 days in normal conditions. Like in a normal lifespan will be 120 days, and if it is a sickle cell anemia, it will be 60 days. So what happened after the 120 days? How it will be? It will be destroyed. Aged RBC, like it going through the spleen, and spleen has a lot of macrophages. So these macrophages are like to kill the pathogen. So for that it needs the eat me signal. You destroy one. This is a membrane structure. So these are the phospholipids are there, and that will be, you can imagine, that's a protein, transmembrane protein. So it's a membrane. Let's say it's a RBC membrane. So the membrane has the different kind of phospholipid. So one of the special phospholipid is the phosphatidylserine (PS). Let's say that is the phosphatidylserine. So phosphatidylserines normally it is in specific layer. In that layer it will be present. So what happened? From this side to this side, from the lower side, like this in that layer is a cytoplasm. So it's a cytoplasm. So and this is the ECM network, extracellular matrix. So it's like the flip out that to the top layer, and this is called the eat me signal. If it is there, they are phosphatidylserine flip out. Macrophages coming through the phagocytosis process. So it's engulfs, and after that macrophages coming through the phagocytosis process. So it's engulfs, and after that it's breakdown to two. One component is the hemoglobin. Hemoglobin we get, and then it disrupted into the porphyrin ring and the ferrocyan. So like nothing will be waste. Like the Fe will not lose, so no iron loss. So another thing it will happen, globin protein is there. Globin is there. So globin protein is there. So and then globin to the amino acid. So globin to amino acid. Porphyrin ring, it's converted into the biliverdin. Biliverdin is kind of green molecule. So then it get converted into the bilirubin. Bilirubin is you say it's yellow color. So it's the bilirubin is a yellow color molecule, and this bilirubin going to the liver. Now let's going through the bilirubin part. So already we have seen bilirubin is there. So this bilirubin going to that liver, coming to liver. Coming to liver. So liver has a specific enzyme that is called the UDP glucuronide transferase. So write down UDP
glucuronide transferase. So
like this enzyme is there in that liver. So this bilirubin in liver, it gets changed the bilirubin plus glucuronic acid, that is the conjugated bilirubin. So it converted into, like one portion is like bilirubin, that is unconjugated one, not conjugated, plus one will be the glucuronic acid. So some will bind with the bilirubin and gluconic acid, and it will form the conjugated bilirubin. So that conjugated bilirubin stored in the gallbladder, stored in gallbladder. So, and after some times it can release into the intestine. So that is the release to intestine. So that means in summary, the RBC get breakdown and it converted into hemoglobin and the globin, and some other portion it is a lipid or protein part that can directly coming through the amino acid section. So in section there the porphyrin and Fe2 plus it's converted in the biliverdin, and it's kind of green color, and the bilirubin that's a yellow color, and then it will be coming through the liver, bilirubin coming to the liver. Some enzymes is there like UDP and glucuronide transferase, and this enzyme binds with the bilirubin and form the glucuronic acid, and that is also known as a conjugated bilirubin, and that's stored in the gallbladder, and that releasing to the intestine. So what happened? Uh, some interesting microorganisms, few of the microorganisms are there in the intestine, specifically in the large intestine and small, but in large intestines a lot of microorganisms are there. This microorganism, like I can write down the microorganism. So this that converted the conjugated bilirubin to the stereobilin, and in that stereobilin form it is get excreted out. So that is the complete destruction, and how the iron get recycled, that we will discuss in the next slide. Okay. Now coming to that, how iron is get recycled. So already we have discussed about the porphyrin ring, and so let's write down once. Porphyrin IX plus Fe2 plus. Now Fe2 plus binds the apotransferrin. So iron Fe2 plus apotransferrin. So that form ferrotransferrin. Fe or ferro, and this ferrotransferrin coming back to the liver, and this transported to the liver, and then there, so transport of the heme protein. So ultimately what happened, they are one kind of heme is transported to the liver, that is the known as hemopexin. So you can write down the term, what is the hemopexin. Hemopexin, it means the transport of the heme to liver, that is the hemopexin, the transport of that. So in iron transport, apotransferrin is another protein that involved, and there is a ferrotransferrin, and this form of can going to the liver. So already we have discussed how the yeast RBC get the elements of that is RBC, how they recycled and how it is formed their intestine, and how it is coming through the intestine through the bilirubin and biliverdin, and that is the important parameter when we get infected in some disease or the RBC breakdown, and at that time the role for diagnosis purpose bilirubin and biliverdin parameters are getting checked. So let's talk about few random things that kind of related to that. So what you can also write down the random thing like ceruplasmin. One of the key, ceruplasmin. This is kind of random, you can say, but related to it. The ceruplasmin is that normally we see the in hemoglobin the Fe ions are there, like Fe2 plus are there, but if the copper carrying protein is there and remove the iron from the cell, that is also known as the ceruplasmin. So you just remind the copper ion here instead of ferrous, or there's a ferrous. Now if the iron deficiency, another key term, if the iron deficiency is there in that cell, like the microcytic anemia, it is also known as their hypochromatic anemia. Like iron has the role in their hemoglobin synthesis. Like Fe is the Fe lack, then what happened? That is the hypochromatic anemia. This is a disease condition, hypochromatic anemia. This is a hypochromatic anemia, and this kind of thing, like iron loss happen on that kind of thing, and that has the important parameters, like if I say it's a decrease, then the Hb production, hemoglobin production, get hampered, because in Hb synthesis regulation, iron has HRI complex, or you can say. So the another important key factor is the HRI kinase that involve the heme regulating inhibitor kinase, heme regulating inhibitor kinase. So like if heme is increasing, then that inhibited HRI kinase. So you can say the right term, that is it inhibited the globin synthesis. Globin synthesis. So like if you heme or Fe2 plus, if Fe2 plus get decreased, their heme getting decreased. If heme is decreasing, that means HRA kinase getting increased, like it cannot inhibit reciprocal condition of that, and the globin will decrease. So that kind of regulatory mechanism that found in the hypochromic anemia. Hope that everyone get understand with that specific topic.